Thanks, Eric. I am not a high wire gymnast, but could have made something up, and you can train me. All right, so I have to point at.
Yeah, I think the battery just wants to get close to that little thing. Sometimes it seems there is.
Can I just use the laptop?
Yeah, if you want. Why this is seating? Just it seems like it doesn't want to work first, and then it starts working.
Escape and then reopen it. So something between when you share and when you go into presentation mode.
Okay, now it's working. There we go. Okay, so try now. Okay.
Yeah, there you go. Okay. A couple important disclosures. I know this isn't a CME. I get a lot of support from sequencing companies. The data that I show will be data that was generated with their support. Okay, so as Eric mentioned, my lab, we do a lot of long-read sequencing, and essentially the goal is just to improve diagnostics for rare and common conditions. As a clinical geneticist, I see kids in clinic about 30% of the time, so I think about to see with them, and really what I want to get to is getting to a diagnosis more quickly, and hopefully someday a diagnosis that also has additional prognostic information. I think this is becoming more and more critical, just as we're seeing that therapies, as straightforward as just getting PT/OT early in life make a big difference, but also as new treatments come online, like we heard about yesterday, and then also some really exciting stuff I saw last week at the Angelman meeting. Okay, so we use long-read sequencing. What does that mean really quickly? So long reads are longer than short reads. That's the simple answer. The easiest way to think about it is within any repetitive region, these can be complex or these can be difficult to evaluate using short-read sequencing. Essentially, you may not know exactly how many repeats are in this region, and even if you can accurately determine the number of repeats, you might miss different that can be clinically meaningful. Long reads are simply, most of the time, span the length of the repetitive region themselves. So you don't have to go through the business of reconstructing this and then asking yourself, did I accurately, or did I, how confident am I that I identified some sequence difference? If you want to put a few numbers on it, it's pretty dramatic how the cost has fallen and accuracy has increased over the last few years for long reads. So of course, short reads, you're talking, 100, 300 base pairs, somewhere in that range, highly accurate. And of course, to sequence one of us, it's about $500 US dollars or less. I say long reads is anything from 1,000 base pairs up to several megabases. Both Nanopore and PacBio are now above 99% accuracy. PacBio is a little bit more accurate, but they're both useful clinically. And then I think what's most impressive is that the cost has fallen dramatically for both technologies. If you listen to the PacBio earnings call the other day, they talked about the $250 long-read genome coming next year. So we're really at a point where it's not a cost difference in data production. It's kind of a platform analysis, like, are you ready to generate this data at your institution? And then relevant to today's talk, these are information-rich reads. So it's not just that you're capturing the A, G, C's, and T's, you get the modifications and other things. So as I mentioned, two technologies, just really quickly, the way PacBio works, you're making a movie of a polymerase as it replicates a circular molecule of DNA. You're watching fluorescent molecules come in, so you know which sequence is being added. For methylation, it's a kinetic question, whereas a modified base takes slightly longer to go through that polymerase than an unmodified base, and that's how you can determine CpG methylation and 6-MA modifications. These are highly accurate reads, as I mentioned, and the reason is that you're reading the same DNA molecule multiple times, so any error that you come across gets kind of washed out over the other reads. You are size limited in a way here, where you're really sequencing things that are about 20,000 base pairs. Nanopore works differently. I just have to make this movie work. So you're, oh shoot, I went through all that and then I hit the wrong button. Okay, thank you. So you're detecting changes in current as a single-stranded molecule, which can be DNA, RNA, or protein, passes through a protein pore. So these current changes are characteristic backwards to figure out which nucleotides or amino acids are in the pore at any given time. The nice thing is you can basically detect any modification you want, so any DNA modification, and then of course any RNA modification, which I think is an exciting future application of this technology. relevant to kind of the poly-A tail link discussion that we had yesterday, you can also, when you're sequencing native mRNA, you can actually detect that poly-A tail link very accurately, which is another neat application here. The problem with nanopore, as many people know, is that working backwards from the current to the actual sequence is tricky, computationally expensive, and it is error prone, especially within a homopolymer. Out though that PacBio has a homopolymer error rate as well. And then just one quick note, there is Illumina 5 bases coming. Give up, which I think is good for all of us because innovation benefits everybody. So essentially, native detection, not native, I'm sorry, but detection of modified bases on their existing platforms were essentially, and this is all stuff you can find online, you just kind of go through a typical Illumina library prep. There is an additional enzymatic step that converts modified bases, and then you just kind of continue on, and then within the Dragon pipeline, it'll tell you which bases were modified and which were not. So we're generating data on all 3 platforms for evaluating methylation signatures. Just given the time, I'll be a little quick on this slide. I just want to point out that most of the historical concerns about long-read sequencing, I think, have been resolved. Again, cost is, the error rate is no longer really an issue for both Nanopore or PacBio. The tools to analyze the data are still complex, especially the tertiary analysis tool. I haven't seen one that I really trust or that I like. They're all just repurposed short read tools at this point still. And then you can basically sequence any sample source you want for long read sequencing. So we often do DNA from urine, from dried blood spots, saliva, et cetera. Okay, so with long reads, we can evaluate different variant types, and I'm going to focus on methylation. There's a lot of, you can kind of continue down this path of what is it you're looking at with methylation, imprinting conditions like Prader-Willi-Angelman, X inactivation, of course, but today I'm going to focus on methylation signatures or epigenetic signatures. So what are these? So this is a figure from a paper from 2021. We've seen Bekim Sadovic's name a couple of times. And this kind of nicely shows the power, but also I think the challenges with methylation signatures. So on the left, you have a heat map. You have you have a set of controls, which are in green, and individuals with a condition, the DeLange, so they're in blue. And you can see that they nicely separate. The y-axis is just the CPG sites that are used to group these individuals. Really nice separation of cases from controls. And the point or the idea here is to show you that you have 2 individuals with an uncertain variant, and using methylation alone, you can group them. So this individual would group with controls, so you might say that this variant isn't actually pathogenic. And then you see this individual over here grouping with the cases, so you can say that that individual probably has a condition that that variant is pathogenic. You can represent data like this in multiple ways. So Anna showed some nice, not PCA plots, UMaps, thank you. This is a PCA plot. And here, this is just the same data represented from the heat map. And the idea here is that you're supposed to draw kind of this mental line that says, oh, this is separating the cases from the controls. And here's your 2 red dots. Every time I look at these, the question I always ask myself is, I want to understand why are these 2 dots closer together, and why is this dot and this dot further apart? Dr. Harris yesterday, showed that really nice example of the family with the EHMT1 deletion, where the individual was kind of separated a bit from the group, and really what I enjoy, what is it that's really driving that separation? The challenge is, how do you get the data that's been shown to actually work with it in your own hands? And that is the inherent problem with epi signatures or methylation signatures today. So my question to you, and I'm happy to hear arguments against this, is can you get the data to recapitulate any results that have been published? And kind of continuing on that line, how could you actually incorporate this data into your own research? So what I've found is that typically probe sets, they're sometimes published, so you could go back and you could say, okay, I know which y-axis. So they might just show you the top like 10,000 or so differentially methylated probes, and it might just be in a table somewhere. The classifier is kind of almost never shared, so these SVM models, what you kind of built the model on is usually not shared. And then the reference data or the control data are essentially never shared. The This is kind of a, I guess, a direct criticism, but it was a really nice example that popped up last night while I was finishing my slides. I was on LinkedIn, wasting time as I sometimes do, and I saw this post. Literally, this was last night. So just out in Genetics and Medicine Open. So this is the IDefine group up in Canada. They evaluated a condition called POGZ-related disorder, and it's White Sutton syndrome, and they talk about how they can use, they were able to use epicine or methylation to help classify individuals with this condition. Okay, so go to the paper, and here's the data availability statement, and this is basically the exact same data availability statement that's in every one of their papers. So the individual genomic and epigenomic data are in the database are prohibited from deposition in publicly accessible databases due to institutional ethics restrictions. You can't ever get the data. That's the general problem. I do want to take a second and point out that, so we heard from some folks in Dr. Wexberg's lab yesterday with Dr. Harris's presentation that the data that's on Epigen Central, which Dr. Wexberg's host, this is an excellent resource signatures and understanding them, and I would point you to this, of course. And then I just want to show that they do link to some publicly available data that they've generated, which is available on Geo. And this is kind of the source that I use to learn how to work with this data, so I do really appreciate that. So what's my goal? Why am I standing here? Why do I go around the country collecting samples from kids with rare genetic conditions? I want to build a resource that enables research for everybody, not just the groups that have the samples. I want to make sure that the underlying data is available for everybody to investigate, to look at, to understand what's driving the signatures in their condition. And that really helps us understand the impact of the variables that you can modify when you're making methylation signatures. So using long-read sequencing, and I didn't really mention this at the beginning, we can evaluate all 33 million CPGs within our genome. You can do the same thing with Illumina, of course, but you get poor resolution within the complex regions. Arrays, which most of these signatures are built on, only evaluate about 900,000 CPG sites. So we can see a lot more modifications than we can see using arrays. And then, of course, we want to build and release tools that can be run locally so that you can run them in your lab and at larger scale. We think this is going to dramatically improve the clinical impact of methylation signatures. Dr. Harris, again, yesterday, the really nice example of you had a discordant phenotype, right? You had an individual with the genetic change that, trying to understand why that individual seemed unaffected, whereas they may have had an affected child. So getting access to the data helps us understand that. It helps us improve our ability to predict the impact of specific genotypes. And then I think this is really important in the coming years. Can we develop ways to objectively measure biomarkers that are telling us what the response is to a therapy? Gene therapies that are coming, even small molecule treatment, PTOT, things like that. Can you actually see a difference in methylation signatures in individuals? This is a plot. So Sophie Gibson's a PhD student in my lab. She's done a lot of this work. She's really amazing and has really driven this project. This is a gene we've been looking at in a particular condition. This is the 5' UTR, the first exon. For this condition, this is the most differentially methylated site within the genome. So what you have here in this like salmon orangey color is affected individuals, and then the dark line is the healthy controls. You can see that the methylation fraction, which is on the y-axis, is pretty similar throughout most of the gene, except for at this highlighted region, and then a little bit here before this kind of arbitrary 50. If you look at the microarray, the CPG sites that are evaluated all fall within this region. So you're really not able to capture the difference in methylation here using an array-based approach. So you can really only see this using either long-read sequencing or short-read sequencing when you can look across the entire genome. So this really highlights to us the importance of moving away from array-based approaches and moving toward more whole genome approaches for this condition and for probably most others. We have been able to use publicly available array data to recapitulate some of these methylation signatures. So here I'm showing you data from controls in green, Kabuki syndrome in blue. This is array-based CPG, or I'm sorry, publicly available CPG sites. So we knew where to look, and then this is array-based data, so we could just recreate this ourselves, and you can see nice separation on PC1 for these two conditions. I'm sorry, for this one condition from controls. When we overlay our long-read data, we see that it does group nicely using those published CPG sites. So we have two individuals with Kabuki rate sequencing on. They're the two blue triangles up here, and then we have a set of controls here as well. You'll also notice that there's along PC2, and this is going to be technology specific probably, right? So this is because these are ONT derived, these are nanopore derived, whereas these are the array-based ones. Things can get a little bit more challenging with other conditions. So here's charge syndrome. Again, publicly available data, publicly reported CPG sites. Individuals with charge are pink. The controls, again, are green. And you can see that we do get good separation between cases and controls. I would point out that there's this one pink dot that's closer to the green. I'd like to understand why that is. So this is, the whole reason I'm doing this is to get at what makes those two dots different. But again, we can include our long-read data, and we see nice separation here. We've built our first long-read derived epi-signature from a condition for which no publicly available data, there is no publicly available data. This is set D1B. This is work Sophie's done again. So here you see really, really nice separation between controls and cases. We've had several groups now generate their own long-read data, plug it into EpiSignaler, which is the tool Sophie has written and that we're going to maintain long-term for this. It's available on GitHub, and it correctly classifies their cases and controls. So hopefully our manuscript on this will be out in a month or two, our preprint at least. But we've been really happy about how nicely we can separate using just the long-read data. It can get pretty tricky depending on the condition. I think there's going to be easier conditions, if you will, and more difficult ones. So set D1B seems to be a bit, quote, easier. I think Kabuki is probably a bit, quote, easier. Charge might be a bit more difficult. I don't know how to quantify easy and difficult right now. But this is Kleefstra syndrome. This is publicly available CPG sites and then publicly available array data. The orange is Kleefstra and the blue are controls. Sorry, the color. Either way, separation between cases and controls here. When we overlay our long-read data, we basically see no separation. So we're thinking that whatever the CPG sites that were used to separate the array-based data aren't going to work for the long-read data. We did try to redo these signatures using just the array-based data, building our own, essentially building our own signature using the array data. We weren't able to recapitulate this result here on the left, which is okay. I mean, there's just a lot of things that go into developing these. We have worked on a long-read specific Kleefstra signature. We get nice separation here. I'll say I'm not super pleased with this though for a couple of reasons. First, the controls spread out a bit, and we have had some challenges when we add additional individuals that weren't in the training set, they don't cluster very well. So we're trying to understand this a bit more. This is just to say that Kleefstra, I think, is going to be a bit of a difficult one to work with. And then my last slide, and this is the thing that I think is just really highlights the need for doing this across different technologies. Here's Kleefstra syndrome, individuals that are sequenced on both the PacBio and the Nanopore platform. And you can see that we get nice separation of PacBio from Nanopore, from controls, but we need to understand why are these two different? When we're looking at the same CPG sites, we know this is going to be technology specific, but why aren't they measuring the CPGs in the same way? I do think, at least for the foreseeable future, we'll have to generate all of our controls across all platforms and then do platform-specific evaluation until we really can understand how best to control for these platform-specific differences. Okay, so I got some dried blood spot cards from Combined Brain, so thank you, Terry Jo. We haven't sequenced them yet. We need to talk about the quality of those. But I've got some for Kleefstra syndrome. They're kind of going through the queue right now. my goal here at this meeting is to actually collect blood in person, both today and tomorrow. I do it using a remote blood collection device called a Tasso. It's kind of a lead arm, push a button, and it pulls out about 1/2 ml of blood. We tend to get about 5 or so micrograms of DNA out of that, which works really well for, we can sequence it across all 3 platforms. So we generate that data. And then I am looking, kind of as Anna was talking about in her talk, recollecting from the same individuals over time and seeing how those signatures change with age, because I think it'll help us understand kind of some of the responses that people have to treatment. With that, I'll just stop there. I'll say thank you. Thank you for the opportunity to come and talk about the work. And then, of course, thank you to the community for letting us generate this resource.